Certainly, but first of all, I want to say I'm delighted to be here. I'm actually really impressed by the group you've put together at University of Wisconsin, so I'm very happy to be talking to you guys now. About my background, so I'm actually an economist by training. I did my undergrad in mathematics, but then did my PhD in economics. I was too fascinated by what was going on in genetics to stick to economics only. So I've been trying to bridge the gap ever since, well, with a few, not a very linear path. But anyways, so I'm now at the Interdisciplinary Center for Economic Science and at the Department of Economics at George Mason University. And the paper I'll be talking about is entitled Nature and Nurture, Evidence from Molecular Genetics Data and Adoptees. And so this is joint work with Lauren, who's here. And Lauren, please, if I say anything that's wrong or anything, please do chime in. Although I think I should get it. I think I'm on top of this. But and this is joint work also with James Lee and Matt McGew, mainly worked with the data. Okay, so without further ado, I'll get going. So, oops. Okay. Well, I have to say I've used this slide deck to talk to social scientists and economists. So some of this is more, I've done some edits for this crowd, but it's still, some of this is geared towards economists. So, and some of this is obvious to you here. So nature and nurture debate has long captivated humankind. And over the past 50 years, twin and pedigree studies, such as those by Behrman and Taubman and Cesarini and many others in economics, and adoption studies, such as the one by Beauchamp et al. I don't know if you know these papers in economics, for those who are not economists, but the point here is there's a whole behavioral genetics literature in economics that looks at the nature nurture or the genetics and environmental factors of quote unquote economic traits. However, as is well known, these studies are limited for various reasons. First, they require lots of structural assumptions, but in particular, it's very difficult to quantify the influence of nature and nurture in the interactions. And one of the recurrent problems is selective placement in adoption studies. So meaning that adoptees tend to be placed not randomly in any family, but they tend to be placed in families that are more similar to their biological family. So they're not just randomly allocated. So this is a well-documented issue. So whenever you look at ACE model or a regression coefficient that regresses an adoptee outcome on adoptive parent's outcome or variable, there's always the risk that there's omitted variable bias, meaning that the adoptive parents are similar to the biological parents, which in turn transmit their genes to the adoptee. But in a pioneering study, Bruce Sacerdote, who's an economist, studied a set of adoptees who were quasi-randomly matched to the adoptive families. So what he did basically is he used a set of adoptees of Korean origins, and for them, the adoption process was such that the adoptive families has really literally zero information on who they're getting. And there was I'll talk a bit more about this because we do something similar basically. At the same time, so that's for the study of nature nurture and old pedigree data, so to speak. At the same time, as you all know here, technological advances now make it possible to work directly with molecular genetic data. And in this paper, we combine both features, basically. So we analyze a unique data set in which adoptees were quasi-randomly matched to their adoptive families, and in which individuals have been genotypes. So we kind of follow in the footstep of Sacerdote in studying quasi-randomly matched adoptees, and then we also get molecular genetic data for these adoptees. Okay. Oops, just a second. Okay. So quick overview of the results. So we compare the resemblance of adoptive versus non-adoptive siblings using standard behavioral genetic methodology. And doing this, we find evidence that both genes, meaning nature, loosely speaking here, and the environment, meaning nurture, loosely speaking, matter for cognitive and non-cognitive skills, BMI, height, and smoking. Okay, no surprise here for anyone in this Zoom room, I'd say. But that's just to set the stage. Then we leverage the molecular genetic data, and there we also find that both genes and family environment variables impact adoptee outcomes. So all studied outcomes are significantly impacted by genes, and consistent with the behavioral genetics literature, family environment variables have relatively small effects on adoptee outcomes and are often insignificant. There's a big caveat here. I'm not saying family environment doesn't matter. I'm saying in those data and with the family environmental variables we look at, we don't find huge effects, but there's still effects. Okay, so it's important to qualify that here. I should also say, I should have added a bullet. One thing I'll spend some time talking about is genetic nurture. So one unique feature, like the unique feature of this data set, is the quasi-random placement, which makes it perfect to look at genetic nurture effects. And so we have some interesting stuff to report on that, but I'll get to that in due time. And then the third big piece is we find suggestive but surprisingly robust evidence that genes for EA, quote unquote here, meaning a score for apologetic score for EA, and family socioeconomic status are substitutes in the production function of cognitive skills. So that's the economic jargon here. But the point is we find a negative gene-environment interaction between the score of EA and family socioeconomic status. And that's surprisingly robust. And however, we emphasize that the statistical power is limited. We have a small data set, and in fact, we've looked a bit more formally in the issue of power, and I'll comment on that. So replication is needed. So we take this result with a grain of salt until it's replicated or not. All right. So for the rest of the presentation, actually, I should have asked at the outset. I have until 11:30. Is that correct? And are there question period at the end or questions during?
That's our fault. It's up to you. If you want to set the rules, we'll abide by whichever ones you'd like.
Okay, so I can take questions whenever, and if ever there are too many questions at some point, then I'll just tell you and keep going, and then we can take more questions. Okay, so if you have any questions, go for it. Okay, so for the rest of today of the presentation, I'll first talk about data. Then I talk about nature and nurture with the evidence from pedigree data, so old-style behavioral genetics. Then I'll talk about the evidence from molecular genetic data, and then I'll talk about the interactions between nurture and nature, or G by E. So starting with the data. So the data is from the Minnesota Center for Twin and Family Research, or MCTFR. It's data on 409 adoptive and 208 non-adoptive families, and each of which consists of a pair of siblings and their parents. So many of these families, all these families had at least 2 children because that's the way they were sampled. Some of these had more, but those with more than 2 children, only 2 of these were sampled. So in this data in particular, there are 421 Korean adoptees, including 361 who were genotyped, and 471 white non-adoptees, including 411 who were genotyped. And by white non-adoptees, I just mean children with their biological parents, basically. And then for all of these, we have data on their adoptive parents who happen to be all white. Actually, there were, I think, let's say like 3 or 4 who were not white, and we decided to exclude them from the sample just to avoid having to deal with stratification issues and stuff. So for our data set, they're all white adoptive parents. And the data were collected in 3 waves, where the adoptees or non-adoptees were 15 on average, with a range of 10 to 21. Then the second wave, they were 18 years old, and then the third wave, they were 22 years old, with a range of 19 to 28. So 2 things to note. First, the measurement was when they were still pretty young, and many of the data we have are from the first wave. So for instance, the cognitive performance data comes from wave 1 when they were 15 years old with a range of 10 to 21. So, whether our results generalize to cognitive performance in adulthood, they may, I suspect they do in some loose form, but they may not be exactly the same if with adult cognitive performance. The second thing to note is there's a 4th wave of which we're expecting the data soon, some variables, which has data from these adoptees or non-adoptees at an older age when they've completed education. We also should have income data, employment data, and so we're really much looking forward to get such data. Okay, now the unique feature of this data set is that the placement of the adoptees in the adoptive families is effectively random. The agencies matched the next eligible family with the next eligible adoptees. That's the way it worked. Usually, when you don't deal with foreign adoptees, or at least Korean adoptees, I'm not an expert on the topic, but my understanding is that social workers or adoptee placement people try to match the adoptees to a family that's not too different from their biological families. Often it's relatives, but even when it's not relatives, they'll try to match the adoptees in a family that's relatively similar. So that obviously creates a huge issue for people using normal adoption studies, which is that there's a correlation between the adoptive parents' genes and the adoptees' genes so here we should not have that in principle, because the way the Korean placement worked is there was an orphanage or a set of orphanage in Korea, and like the Korean government had a list of Koreans waiting to be adopted, and then the next one on the list would be matched to the. On the other end, in the U.S., you had in Minnesota, you had families who were on the list to adopt, and it was queues, and then basically the person who was at the top of the queue in the U.S. got matched to the adoptee that was on the top of the queue in Korea, and so on. So families, at my understanding, could express a small preference for sex, but that's pretty much it. And even then, it could not always be respected, if I understand well. So basically, there was very little scope for parents to. There was very little scope for selective placement, basically. The second thing is we did a statistical test to see whether the variables in the adoptees that were predetermined, meaning like the scores, we treat height as a predetermined variable, although you can argue, placement, age, and many other adoptive variables like that. We look for their correlations with the family variables, such as family SES, whether in a rural or urban area, and so on, and we find no correlation whatsoever. So we feel pretty confident that this quasi-random placement of adoptee. Now, the main adoptee outcome are cognitive and non-cognitive skills. Going to college, non-cognitive skills is basically an index of soft skills. That's based on, actually I have to look again at the precise definition, but it's basically on motivation and good behavior and working and so on. Then there's a dummy for going to college. I should say this is not a perfect variable because some of these adoptees had not completed college, because as you can see, they were 1928. So for those who are in college but have not completed it, we code this dummy to 1. So it's not the same as exactly as completed college. It's kind of a measure of college attendance or having completed college. High school GPA, body mass index, height, and ever smoked. And we're also waiting for data on cigarettes per day and drinks per week. And as I said, from wave 4, we'll get some more data on income, employment, and so on. And the main experience variables of interest are the polygenic indices. I'm sorry if I switch between polygenic score and indices. This is kind of the new terminology that we use in some work. But so in this presentation, I think I use both, which is quite confusing. So polygenic scores and polygenic PGS or PGIs are the same. Another key variable is family SES, which is based on parental income, education, and occupation. So it's a score used by sociologists that were constructed. It was constructed for us, and we're just using it. It's apparently a very commonly used index of family SES and other family variables. Now, the control variables that we use, we have a baseline set of control variables that we use in pretty much all regressions, which are sex, age at measurement of the traits. So if, let's say, IQ was or cognitive performance was measured at intake, we have the age will be somewhere between 10 and 21, so control for that. Placement age and month, so that's the age at which the adoptees were placed in the family or got into the adoptive families, and these are all between 0 and 24 months. So all adoptees were adopted before 2 years old. Then we also control for adoptee birth year and the top 10 pieces of the genetic data, and these were controlled separately for the Koreans and for the whites. We obviously control for the appropriate PCs in both cases. All right, a polygenic score.
I have a quick question.
Yes, please.
So can you elaborate a little more why the non-random placement of the children and the rearing parents would be problematic? So shouldn't that, as long as their genetics are independent, will be fine?
Yes, and that's the last point you've made is key. As long as their genetics is independent, will be fine. But suppose that but when there's non-random placement of adoptees, the genetics will probably not be independent, because suppose an adoptee is placed in an adoptive family that's similar to its biological family, then suppose the adoptee comes from a high education family, biological family, and is placed in a high education adoptive family. So then the adoptees in expectation will have a higher than average polygenic score for education, and so will the adoptive family. So that's the confound here.
Jonathan, I have a quick question.
Mm-hmm.
Does it matter that, like, it could be that the Korean adoptees are just drawn from kind of a different distribution of families than, even though they're randomly assigned to American families, than sort of the kids in the American families? Does that cause any issues? Like, is that a problem?
Yeah, it's a good question, and unfortunately, the answer is we don't know. So sorry, I think I put this in the way. Unfortunately, we don't know. So that's a very good point. So the key assumption, well, you can see this work in two ways. First, you can see as this is in sample, or like it's applicable, generalizable to a sample of Korean adoptees in the US, which obviously is not that interesting because most people don't fall in that category. The other interpretation is that you generalize this to the general population under the assumptions that indeed whatever happens with Korean adoptees is, that there's like parents don't treat Korean adoptees differently than they would their own children or white adoptees. And so, I mean, by the way, that's always a concern with adoption studies. What if parents who adopt treat adoptees like differently and are not, like, results are not generalizable? Here it's even more so, because what if there's the racial angles, like what if parents treat Korean adoptees differently for whatever reason, So yeah, we have to assume, to generalize the results to the general population, we simply have to assume that that this is not an issue, basically. And yeah.
I have a minor question, Jonathan, about the data. Is it by design that each family has two children, some of which have one adoptee and one non-adoptee? Was that just the way the data were collected, or are there a set of families with only adopted children but are excluded from the analysis?
Yeah, so the way the data was collected is all families needed to have at least two children, and then they only measured the data for two of these. So some of these families have more than two, and some of the families with two children that are in the data are two adoptees, some are two biological children, and some are mixed families. Does that answer the question?
It does, and in particular, families with a single adopted child just cannot be in the survey. That's
correct. So this comes from the SIBS study from the Minnesota MCTFR, Minnesota Study for Twin and something research. Anyways, so that data set was meant to sample particularly. So I'm just basically particularly to sample a set of siblings, basically. Yeah.
So really quick, one more question. Mm-hmm. Like, going back to the idea of like, so you were talking about, like, maybe adoptees are just sort of treated different than non-adoptees. But my question is kind of like, so what if in Korea there's negative selection into being sort of put up for adoption? Mm-hmm. So even though that, the subset of people who are, put up for adoption in Korea are randomly assigned to families within the US, like the mean or variance of the sort of, different sorts of polygenic scores of the Korean adoptees is different than the mean or variance of the sort of polygenic scores for the sort of like the non-adoptees. Do you think, does that cause any issues?
It could. So, well, in terms of the mean, I'd say I doubt they're very negatively selected, simply because they score really high on the IQ measure. Although, there's probably positive treatment effect of being adopted in a high SES adoptive families. But but yeah, the range, there's issues of range restrictions both on the adoptee side, although I don't think this should bias the results for the adoptee side, unless we have a model where the effects, the model, unless the coefficient, the model looks different in people that are selected like these adoptees versus in the general Korean population. I think a related issue is range restriction in the adoptive families, where to be an adoptive family, you need to have you need to have like basically you need to be vetted by social workers, and there's some in the placement at the adoption agency. So not everyone can just adopt. So typically they always have, back in the days, there were always married families. They had jobs, at least one job, like they had enough income to have an adoptee. So Matt McGew did a paper on this, actually comparing the summary statistics for like income, education, a bunch of things, and actually a whole bunch of things for the sample of adoptive parents in the SIB study that we use, to those of the census in Minnesota. And he found actually the amount of range restriction is, there's clearly range restrictions. I forget the precise number, but I think he found like for the variance of education attainment is something like 70% of the variance you see in general population. The variance of income is also something around there. For disinhibition and alcohol use, it's also around there. So you do get range restriction, but it's not as overwhelming as some could think. But that's also an issue to keep in mind. This does not cover, the adoptive families do not cover the extreme poverty nor the extreme wealth that we see in the US. It's typically middle, I mean, from very lower middle class to very upper middle class families, let's say, that we get in the sample.
Yeah, just a quick follow-up.
Yep.
So I wonder what's the rationale of focusing on families with Korean adoptees specifically? Because first thing is, I would assume that maybe the families with white adoptees will have a larger sample size. And then second, if you eventually plan to use molecular genetic data, wouldn't that be a problem if the polygenic scores on Asian samples are just not as predictive?
Yeah, absolutely. So these are two good points. So the reason we use Korean adoptees is simply because that's the only data set in the world we know of that has genetic data on quasi-randomly allocated adoptees. So if you were to use white adoptees, there's this issue of selective placement where that I've been talking about earlier, that the adoptive family is characteristics are correlated with the biological family's characteristics, and thus, there's a genetic correlation between the adoptive parents and the adoptee. So that's the main reason, is because it's the only data set in the world that has, that we know of at least, as quasi-random allocation of adoptees to their families. Now, in terms of projecting using polygenic scores in Koreans, let me get to that topic in one or two slides. So polygenic scores, I think everyone here knows what it is, so I'll skip that slides. So here are the incremental R-squares of the PGI or PGS of EA, education attainment, cognitive skills, BMI, and height in the MCTFR. So what we did here, let's focus on this, this quadrant here. You see my mouse, right? Someone just confirm you can see my mouse? Yeah, okay, perfect. So here we have, here what we did, Y is the left-hand side here is variable is cognitive skills, and the right-hand side is the PGI cognitive skills. And what we did is we ran the regression of cognitive skills on the baseline controls, which are the PCs, age, placement, age, sex. Now we ran that regression, but we added the PGI of cognitive skills, and we look at the difference in R-square. So in white parents, the PGI of cognitive skills explain nearly 20%, which is huge and bigger than what we typically see. I think this is like the latest best PGI. Well, this PGI we use in any case in the paper was reported is about 10%. So I don't know why it's so high in this data. It could be just noise, but in any case, that's now in the white children, it's around 0.15, which is also higher than you'd expect. The key point here is that in Korean adoptees, it's quite large also. It's at 5%. So, and we have a similar story everywhere. For going to college, the score of EA explains about 9%, which is similar to the literature. So it's just the results here for cognitive skills you look weird because the R-scores are much larger than what's in the literature. But in any case, for college, we see a similar pattern. Again, the PGI of education attainment predicts about 6% of their variance in the Korean adoptees. I should say the blue are the white parents, the orange are the white children, and the gray are the Korean adoptees. So in Korean adoptees, you explain 6% variance of going to college. For height, we explain about 6 or 7%, and for BMI, about 3%. So, there's a whole literature, I'm sure you all know about it, about like the polygenic scores don't predict well in other races or other ethnic group or their ancestries, whatever word you use. And it's indeed the case. However, it's not the case that the predictive power is 0. It still explains something. What's very important is obviously you cannot control the means of the score in different ancestries, because that obviously that raises all type of issues, both technically and otherwise. But in terms of just predicting within the Korean adoptees, this actually does like partly work. And actually one advantage of doing this is that their stratification is really not much of an issue, at least in principle. I think you've probably followed the literature lately about how stratification is a big issue, even in height, with the series of eLives articles by, I think, Berg et al. And some other. So here, because like basically look at the giant GWAS and saw that there's quite some stratification between Northern European and Southern Europeans and so on. But here, when you train a score in Europeans and project in Koreans, I doubt that stratification is a huge issue here. But we still control for the principal components. So I think if I had to say two main limitations of this study, one is the sample size is limited, and the other is this cross-ethnic PGI prediction, although I'm not sure how much of real limitation it is, aside that from the fact that the predictive power is more limited in Koreans versus in than it would be if we had a score trained in Koreans, basically. Okay, now going on to the results. So we'll first talk about the evidence from pedigree data. So this won't be anything too surprising for you in this crowd. But basically, we did in this section, what we did is we looked at the ACE model, which I won't go through the detail. You have y is equal a plus c plus e. Assume that a plus c and e are independent. I think everyone here knows that. So the main point here is we have three moments, which are the variance of the trait y. Then this moment here. Then the second moment is the covariance between the full SIBs in y. Then the third moment is the covariance between the adoptive SIBs in y. And so from these three moments, we can identify the three parameters of sigma a squared, sigma c squared, and sigma e squared. Okay, so it's a classical ACE model variance decomposition. And here's what we get. So let's first focus on this quadrant here, where we have the adoptive sibling correlation and the biological sibling correlation. And here we have different traits. So for cognitive performance, we see, interestingly, a positive adoptive sibling correlation, even though there's quasi-random placement. And so basically, if c squared did not matter at all for cognitive performance, this would be 0 here. But it's significant. So that suggests clear family influences on cognitive performance. The biological sibling correlation is higher, as expected, because here you have the influence of both genes and family environment. Now, for GPA at intake, again, we do see a marginally significant, in this case, sibling correlation and a higher biological correlation. And for non-cognitive skill, likewise, we see adoptive sibling correlation and a higher biological sibling correlation. And actually, we always see a higher biological sibling correlation, suggesting nature plays a role in all of these, or genes play a role in all of these. One thing to note is that the effect of family influence appears quite large for going to college and smoking, ever use nicotine across all waves. Here, the adoptive sibling correlation is around 0.20 for going to college, and around also around 0.20 for smoking. Then we translated these estimates into the ACE model. So the sigma c squared here is directly equal to the adoptive sibling correlation. Just going back to the previous slide here, we have that the covariance between the adoptive sibling is equal to sigma c squared. So, so that's what we see here. Sigma a squared here is the heritability. For cognitive performance, it's rather low. One thing to note, though, is the confidence intervals are huge, obviously, because we have so few. It's a rather small data set. So this is a relatively low heritability estimate for cognitive performance, but with huge standard errors. And also, it's estimated at an age between age ranges, I think 11 to 19 in wave 1 years old. So at that age, heritability is known to be lower for cognitive performance. Reassuringly, for height, we don't find any huge influence of family environment. Sigma c squared is not significant, and heritability of height is here at 65%, but again, with huge standard errors. For BMI, it appears hugely irritable at 72%. However, I emphasize again, this is much higher than what we typically see in the literature. But again, standard errors are huge, so that's not a precise estimate. Okay, so one takeaway I want people to get here. I think this is a group of social science genomics, so maybe that won't come as a surprise. But when I talk to psychologists, that's something they don't get. Is that, you all know Durkheim's 3 laws, and the first law is everything is heritable. Second law is genes matter more than the family environment. And well, that's still the case here. Everything is heritable, okay? Actually, except ever use nicotine. But this, I think, is just because of small sample. I wouldn't make. But anyways, assume everything is almost everything is heritable, and it's also the case that family environment has less of an influence than genes in all cases. No, actually, not in all cases. I'm sorry. For going to college, if we believe these estimates here, there's even more family environment, although with big confidence intervals. The main thing I want you to take for anyone who's a psychologist not used to work with social science data is that the C-squared is not always so small for social science data. So for instance, for things like income, which I don't have here, but we'll have this data later, going to college and smoking, there's a huge family component. And here, of course, there's large confidence. Actually, confidence intervals are not that small for the sigma C-squared, but in any case, this is consistent with previous results in the literature. So that's just something to keep in mind. For social science traits, there's often relatively larger family influence than for psychological traits, and definitely than for physiological traits like height and BMI. Okay, here are the results from Sacerdote. So Sacerdote is that paper I mentioned earlier that used these Korean adoptees in larger samples than ours, but they were not genotyped. And what he plotted here is the adoptive sibling correlation versus the biological sibling correlation. So I just copied this figure from that paper. This is the diagonal axis here. So any point, any dot, any trait that's above the diagonal means that the biological sibling correlation is higher than the adoptive sibling correlation. Any trait that's on the diagonal means the adoptive sibling correlation is the same, which means there's no evidence of nature at all. So meaning, and any adoptive sibling correlation that's greater than 0 suggests there's an influence of family environment. Okay, so the takeaway from Sacerdote's results here is that in all cases, there's family environment because none of these points are at 0. Okay, otherwise they would be all on this line here. The second takeaway is that in all cases, there's also nature, because otherwise they would be on, or genetic influence, otherwise it'd be on the diagonal, except for married. But the third point is for many of these traits, they're not that far from the diagonal, meaning that, meaning that family environment is quite an important influence relative to nature. So in particular for drinks, and in any case, now, here are the results we had. So this is Sacerdote's results for height. This is our results for height. Similar. Remember that we had large confidence intervals. This is our results for going to college. So it's similar to Sacerdote again. If anything, we find even more influence for nurture, but we have huge confidence intervals. So I wouldn't think this. The two results are not significantly different. This is for smoking. As you can see, in both cases, we find quite some important influence of family environment. The adaptive correlation is close to 0.20. And then for BMI, this is where a result is out of whack with huge irritability. But again, the confidence intervals actually are huge for this estimate. Okay, so nothing too surprising for this crowd, I guess, from the previous section, because you're quite familiar with pedigree models and with pedigree data and quote-unquote old-style behavioral genetics. But I think it's good to set the stage and to see that for these social scientific outcomes we're studying, there's quite some evidence from family environment and of course, for genes also. So now that we've done this, let's look at what we can say using molecular genetics data. To set the stage, the parent-child correlation, as you probably all know, parent-child correlations, for instance, in education attainment, have often been used to argue that family environment matters. But a child shares half their genes with each parent, so such correlations are not informative about the role of genetics versus environmental factors in typical data sets without quasi-randomly placed adoptees, that is, right? If you regress child outcome and parent outcome in normal families with biological child, you can't say much about the roles of genetics versus environment. And for that reason, economists and psychologists and behavioral geneticists have used adoption studies to circumvent this limitation But selective placement is still an issue, right? And I think I should mention this is really something that economists have hammered, I've emphasized a lot, because economists are obsessed with identification. They're obsessed with finding a perfect, quasi-natural experiment. A perfect natural experiment or natural quasi-experiment. And that's why Sacerdote came up with this idea of using Korean adoptees that were quasi-randomly placed. Okay, so while estimates of family effects are difficult to obtain without a quasi-random placement of adoptees, it's also the case that typical genetic estimates in the literature are biased, at least if you interpret them as direct effect, because that's called as indirect effects or genetic nurture. So parent-children sharing of genetic materials can also bias simple estimates of the effects of gene on children's outcomes. So for instance, suppose the true model is that the trait of the child is equal to a constant plus beta C times the PGI of the child, plus beta of the mother times PGI of the mother, plus beta of the dad times the PGI of the dad, plus an error term. And if we just regress the child outcome on the child's PGI, well then, if beta M and beta D are not equal to 0, then there will be an omitted variables bias, meaning that beta 1 here cannot be interpreted as the direct effect. So far, most GWAS in the literature, well, actually, I think all published GWAS as far as I know, and most genetic studies using scores just use the simple correlation where they regress someone's outcome on their score. But if you have indirect effect or genetic nurture, that's fine. You just have to be mindful how to interpret this as being not the direct effect beta C, but rather the direct effect plus some correlation contamination from beta M and beta D because of the child's correlation with the correlation between PGI of the child and PGI of the mom and dad. And of course, this is, this might not be very much of an issue for traits like height, or it's an open question. But for traits, social scientific traits, which I think most of us here are very interested in, that's a big issue because indirect effect and genetic nurture could be particularly important for social scientific traits. So here, another advantage of our quasi-random assignment of adoptees, is that we can get unbiased estimates of the effects of the PGI and family variable. So why is that? Well, let's go back to this regression here. Suppose we regress Y of the child on the PGI of the child. Well, here we cannot have this indirect effect issue because the PGI of the child because in this case, the child does not share any genes with his adoptive parents, his adoptive mother or his adoptive father. So in this case, we don't have the issue of indirect effect of genetic nurture. So we can get, in our case, the beta 1 estimate will be an unbiased estimate of beta C, the direct effect. And of course, similarly, when we regress the outcome of the child on the environmental variables of the family, again, these will not be biased because the adoptive family does not share genetic material with the adoptive child due to the quasi-random placement of the adoptees. Okay, so we did 3 exercises here. The first one is to look at the treatment effects from assigning adoptees to different types of families. So we created 3 types of family. There's high education small families, and we also created 3 terciles of the PGI. So we followed Sacerdote and defined the following 3 families. So type 1 families are those in which both parents have 4 years college degrees, and they have 2 or fewer children. Or those that are in the top 20% of the SCS distribution. So these are basically the high education small family or ISCS families. Okay. Type 3 family is the low education, low SES, or low education large families or low SES families. These are the families where neither parent has a 4-year college degree and where they have 3 or more children, or their SES is in the bottom 20%. And then type 2 family is just those in the middle, whether neither type 1 nor type 2. And for the sake of comparison, we also define 3 terciles of each PGI, indicating whether you're in the top tercile, the bottom tercile, or the middle tercile, the second tercile. The top tercile is the highest one. So we just split the PGI distribution in 3, and we created 3 dummies. And then for various adoptee outcome, we estimated the following: regress y, the outcome, on a dummy indicating whether you're in a type 1 family, and a dummy indicating you're a type 2 family. So here the reference category is the type 3 families. So basically, beta 1 gives the treatment effect of being assigned to a type 1 family relative to being assigned to a type 3 family, okay? And beta 2 gives us the treatment effect of being assigned to a type 2 family versus type 3 family, okay? And I should say, these beta 1 and beta 2 can, in principle, be interpreted as causal because of we have really a quasi-experiment, a natural experiment here where we randomly allocated the adoptees to their adoptive families, right? So whether you're type 1 or type 2 or type 3 family is random. And so beta 1 and beta 2 can be interpreted as causal in the adoptees. Similarly, here delta 1 and delta 2 can be interpreted as causal. Well, okay, I have to be careful with language here, but basically, this is the treatment effect, if you want, of being in the third tercile versus being the first terciles for an adoptee, because again, the adoptees don't share any genes with their adoptive parents, so there's no genetic nurture or any such contamination of the results, if you want. Okay.
I have a question, Jonathan.
Yeah.
Maybe it's two, but I'll start with the one that in your previous slide, you were describing the case where, consider the regression, the Y child on PGI child, kind of towards the bottom. And if you were, so I would be in a total agreement that beta 1 there is a great estimate for a SNP. The question I have maybe for you to outline is, as the child SNPs are converted into the PGI, those beta weights are from GWAS that have not separated indirect from direct genetic effects. And I'm wondering what you think about what that does to the interpretation of beta 1. Again, stipulated that if you were running this SNP by SNP, I would have no question.
Yeah.
It's more as you transform it into the PGI.
Yeah, that's a good question. So I think let's assume. I mean, the way I would think about, I think it still works. So the PGI here is, suppose each SNP has a component that's its direct effect and a component that's its indirect effect. And so basically when you create this PGI, you can decompose the PGI into a direct effect and an indirect effect. So basically you can create this PGI of the child. You could write it's equal to the PGI due to direct estimated effect plus the component that's due to the indirect effect plus maybe bias, if you want, right? Now when you regress the Y of the child on the PGI, because of the quasi-random allocation, there won't be any correlation between PGI and V. Although I think you raised a good point. What you'll have instead is attenuation bias. Actually, so that's a fair point. I have not really thought this through. So what you'll have is, because this PGI here, first, this PGI won't be correlated with the V here, because if the V it will be included to whatever is pre-adoption variables in the V, but we assume this is kind of, we abstract away from that. But we won't be included with any environmental variables from the adoptive families that are, it won't be correlated with any environmental variables from the adoptive families that are in the inner term because of the quasi-random allocation. However, that leaves the issue of attenuation bias. So if the PGI we use was uses as a component that's direct effect and a component that's indirect effect, then the regression coefficient beta 1 here will be attenuated because of this indirect effect. So that's a fair point. I'll write this down, actually. I have to think a bit more. Does that answer your question? Do you have any further thoughts on this?
I think that's a good framework. I interpret it as a measurement error framework. I just wonder, so it's like classical white noise. I just wonder if it'd be worth, this is not really just for your paper, this is for any papers that, it'd be for sibling fixed effects papers too, where you're trying to take out all the indirect effects from the polygenic score, but yet the beta weights themselves haven't done that, right? So this is nothing specific to your paper. I just wanted to point this out for how clean the beta 1 regression is.
It's a very good point, actually. I had not really thought this through. So thanks for pointing this out. I wrote a note of it. Yep, good point. Okay, so I think we were here. So for the results of these regressions, let's first look in the bottom panel here where we have the white non-adoptees, where we regress each outcome here. So first there's, I should say, there's 4 quadrants in this table. This quadrant here, then there's this quadrant here, then there's this one and this one. And each quadrant report different set of regressions. So here in the bottom part, we're looking at, and this is the bottom left quadrant here, is in the white non-adoptees, and these are the regressions of the trait, the outcomes, on the family types. Now in the bottom right quadrant, we have the regression of the outcomes on the PGI tercile. Okay, so when you look in the white non-adoptees, unsurprisingly, pretty much everything is very significant. So being a type 1 family is highly correlated with cognitive performance, and being in a type 2 family as well versus type 3. Being in a type 1 family increases the probability of going to college by 0.38, or rather is associated with an increase of 0.38, which is 38%, which is huge, and so on. However, you know, we can take these as at face value. These are just correlations, because we don't know if this is the effect, the environmental effect of the type 1 family, or the fact that type 1 families share their genes with their children, and this affects the outcomes. Similarly, here for the terciles estimates, we have large effects, but again, we can't really make much of it. These are the total effects rather than the direct effect, because again, being a third tercile of the PGS of cognitive performance means that you share those genes with your parents. We create an environment which could conceivably also affect your cognitive performance. So what we do then is we compare these estimates to those in adoptees. So let's look at the top left quadrant here. So these estimates are directly comparable to these in the sense that you know, the type 1 dummy is exactly the same, the way it was defined, and so on. And the difference here between, for instance, this coefficient here and this one here of 4.168 versus 6.385 is that this one here, there's no sharing of gene between the type 1 adoptive families and the adoptee. And so if you take this as this estimate at face value, it suggests that being in a type 1 adoptive family increases your cognitive performance by about 4 IQ points, and increases your probability of going to college by 21%, percentage point rather, which is quite large. And then the estimates for type 2 are not significant, but you know, they're typically between type 1 estimates and 0. They're not significant, I suspect, because of small sample size, but you know, can't say that for sure. However, you don't see here any more treatment effect of type 1 families on GPA, non-cognitive skills, BMI, height, reassuringly. And because if type 1 increased your height massively, I'd be a bit worried. And even smoking, okay? Now, obviously, the fact they're not significant doesn't mean there's no effect, but they're clearly not huge effects, otherwise they would be detected in our sample. Now, the comparison of this top right quadrant to the bottom right quadrant is hazardous. There's 2 differences. First, there's no, as mentioned many times, there's no genetic nurture here because of quasi-random assignment. The second issue, however, is that, as mentioned earlier, the scores were trained in Europeans, and these are score trained in Europeans predicting in Europeans, and these are score trained Europeans predicting in Koreans. So we can't really compare these coefficients and say, oh, this reflects the lack of genetic nurture versus, oh, this reflects the fact that the scores predict in Koreans, and as such, there's attenuation here. So, so I wouldn't compare these 2 quadrants here. Rather, what's interesting is just to compare the right and left quadrant. Clearly, you know, if you want to make a very simple horse race between genetics and family treatment variables, genetics seems to matter a lot. I would caution against doing exactly that because you know, those types could not be the right types. You know, it's the way we define these might not be exactly what matters. It's just the way we just followed the way Cicero did. And the other reason is because, of course, the scores are themselves estimated noisily. So basically, we can do this kind of like half seriously, but we cannot make really a comparison of overall genetic versus family influence based on that. All right, the next exercise is simpler. We simply regress adoptee care outcomes on family adoptee characteristics. So we have 7 regressions here represented by 7 columns in this table. So let's focus on the first one. Here we have cognitive performance at intake. So that means at age about 11 to 19 or so. And we regress cognitive performance in the adoptee on their adoptive parents' socioeconomic status, number of siblings, child is the dummy indicating whether the child is on the adoptee, and so on, and then on their scores. Okay, and well, perhaps surprisingly, we don't find much influence of the family variable, although family SES does have quite an effect on going to college, increases this probability by about 9.4%. Also interesting is that, what did I find actually? There's, wanted to emphasize something, but I forget. Oh yeah, height. Having a taller mother apparently reduces your non-cognitive skills. So I think this is just due to multiple hypothesis testing, but it's hard to know what's going on here. It's barely significant. It's just at the 5% level here. In any case, as expected, we see the scores do predict the various outcomes. So for instance, here we predict with the score of EA, educational attainment would predict cognitive performance, going to college, GPA, non-cognitive skills. BMI score predicts BMI, of course, and height score predicts height. So I think this table is not too revealing in the sense that the scores predict their traits. But what's quite surprising, and I would not have predicted ex ante, is how little the family variables predict those traits here. But again, the caveat is maybe we did not pick the right family variables, and so this is not a proof that family variables don't matter, of course. It's just, I'd say, quite surprising results. Ex ante, I would have expected more of an influence of family SES, and I think if you ask many people who are not familiar with this literature, they would definitely have expected more influence of these family variables and these outcomes. All right, now the third exercise we did is the genetic nurture or indirect genetic effects. So recent research suggests that genetic nurture effects can be quite sizable, accounting for about 20 to 30% of the total effects for EA. Now I'm not talking about the R-square, I'm talking about the actual total effects, like the total effect coefficient beta. However, these effects could also reflect confounding due to population stratification and or associative mating. This is well known and well accepted. But to develop some intuition on what's going on here, suppose the true model is that the outcome of the child is equal to the child's PGI plus the mother's PGI plus the dad's PGI, right? So genetic nurture will happen if beta M and beta D are not equal to 0, right? Well, if there's stratification, then let us express the error term. Let's split it in two terms. One is S for stratification, and that reflects the effect of cultural and environmental variables correlated with the PGIs. Well, then in the sample of non-adoptee, the covariance between the PGI of the mom and dad PGI, the covariance between the PGI of mom and dad and S will not be equal to 0, right? Because there's stratification, by definition, that's what it does. So that means that the estimate of beta M, that should be beta D and beta M, will be biased, right? Because of omitted variable bias. So basically this PGI will be correlated with the error term, and that will bias this estimate. So in a sample of quasi-randomly placed adoptees, however, the covariance between the PGI of the parent and S is equal to 0, of course, because the stratification relates to cultural and environmental variables correlated with the PGI of the adoptee in this case. And so basically this will not be an issue in this case. I'm sorry, I was not clear on this. In the sample of quasi-randomly placed adoptees, S here reflects correlation between U and the Y of the child. And the Y of the child contains the genes of the child, which are not related to the PGI and mom and dad. So basically, in that case, there won't be a covariance between the PGI of the parent and S. So there's no bias. Okay, so let's look at what happens then when we run these regressions, both in the sample of white non-adoptees, which is typically what's done to explore genetic nurture, and in the sample of adoptees. Okay,
can I ask a clarifying question here?
Yes, please.
So when you're talking about parents and kids, I guess I'm just confirming that we're still in the framework. We're not talking about the children of the adoptees, right? We're talking about like the adoptees and then their adoptive parents. Is that true?
Correct. Yeah, I should have clarified. The data we have is always, child is always the adoptee, and the parents are always the adoptee's parents. So if I talk about an individual, I talk about the adoptee. But if I talk about a child, I also talk about the adoptee. But I usually use the word child to contrast them with their parents.
Gotcha. I don't know if I'm convinced yet that the sort of the nurture effects that you estimate then would be not biased by stratification. But you know, we can talk about that more later, I guess.
Okay, great. What do you want to take a minute to talk about it now, or do you think it would be easier to articulate later?
Yeah, sure. I mean, so like, let's just say I'm adopted, and so we have my genotype and my parents' genotype, my adoptive parents' genotype. I mean, the stratification effects on me, let's just say there's like let's just take some big environmental thing. Like, let's just say genes generally are correlated with, like, you know, likelihood of lead exposure, because so genes are correlated with geography, like where sort of a family lives, and then because of that there's differences in some environmental exposure, let's just say like lead, right? So my parents' genes, we're interested in looking at the nurture effect, so the association between my parents' sort of PGS and PGI and my outcome. I mean, they're still going to have all, like, their genes are still correlated with geography, like the geography of where the family exists. And so, you know, any sort of biases that come along with that are also going to be correlated with them.
So, but in this case, we have, so I think in the typical sample of adoptees, I would be correct, but I think in the sample of quasi-randomly placed Korean adoptees, what we have here is that the error term is equal to S plus E. You can decompose it this way. And this S, remember, is part of the error term that explains the Y of the child, right? So this is a stratification. I mean, basically in the sample of adoptees, of Korean adoptees, this S should be basically 0, because except if you're thinking about pre-adoption timeline, but like post-adoption, right. Like if you abstract away from that, the thing is you take the adoptees and then you put them in quasi-random environments, basically. So you break the link between the gene of the child and this part here.
So I think this is where I disagree, is that I think the environments are quasi-random with respect to the child's genotype. So I think you do sort of the direct genetic effects unbiased by stratification in the same way that like sibling models would. But the child, like the child's environment isn't randomly assigned with respect to the parent, right? Like, because we're not randomly assigning parents' genotypes to environments. And so I think you're still going to be, and we can talk about this more later, we have a meeting, but I think, you know, I guess, you know, we'd have to work out the math, but I think you're still biased by stratification. I'd be interested to hear what other people think.
Okay. Yeah, if anyone else wants to chime in, otherwise we can talk more when we meet.
I had one clarification. I thought this is where Sam was going, which is to say that the PGI of child could be correlated with in utero exposures. Is that right? I think this is how you were separating it earlier, Jonathan, that you're on firmer ground when we're talking about post-adoption, but pre-adoption, there could be correlation between child's PGI and other exposures like in utero environments. Is that right?
Yeah, absolutely, and that's actually a good point. I think we should clarify that in the paper, just to have a very clean framework. Yeah, that's correct. We need to clarify precisely what we mean and bring in this pre-adoption versus post-adoption. Yeah. Okay, so putting these questions aside and assuming I'm correct that there's no genetic nurture, well, let's look at these that I'm correct in my interpretation. Let's look at these regression results. So what we have here is the child's outcome. We did this with several outcomes, but I'm presenting it just for cognitive performance, going to college, and height. So we have, let's focus on this set of results here. So we have, for the white non-adoptees, we regress cognitive performance here on the mom's PGS and the dad PGS, okay? And then we did the same thing, but we added the child's PGS. And as you can see, what's interesting here is even after controlling for the child's PGS, you still see that the mom's cognitive performance PGS is significant, which suggests a genetic nurture effect. So that's basically the idea, or an indirect effect, basically. So that's a traditional understanding of genetic nurture. Of course, some people do it with the non-transmitted allele instead, but if you control for the child's PGS, it's the same, right? Because Now, what's interesting is if you do the same thing in the adoptee, in the Korean adoptees, then the mom's PGS is not significant. Obviously, lack of significance, in one small sample is not this positive, but still, in the same similar size small sample in white adoptees, we did find significant. In the white non-adoptees, we did find some significant evidence for genetic nurture. And I should say the difference between these two columns is just that we control for the child's cognitive performance PGS, which should not matter because, in principle, because of quasi-random allocation, the child's cognitive performance PGS is uncorrelated with the adoptive parent's PGS. So indeed, the coefficients here and here and here and here are very similar. And then for going to college, we don't find any evidence of genetic nurture here, nor for height, as you expect for height. But now we did these results again, these regression again, in the sample of only with the mothers instead. Before we had both the dad and the mother. We did it with the mother only, which allows us to increase the sample size by roughly 50% or 40% because many dads have missing data. So here's the typical genetic nurture regression. We regress cognitive performance of the white non-adoptee on the mom's cognitive performance PGS and the child's PGS. And again, here, this coefficient here would typically be taken at face value as evidence of indirect effects, or with the caveat that this could be confounding. But here, when we regress it in the Korean adoptees, again, without the confounding, we don't find any evidence. For going to college, we have a similar story where we find evidence of indirect effect for college in the non-adoptees, but not in the Koreans. And then even for height, interestingly, we find some evidence in direct effect, although it's marginally significant, so I wouldn't make much of it. If there is something, it's probably stratification more than direct effect. Actually, that's kind of my interpretation of these results, is this is probably really, on the whole, the main point is that I think our conclusion is that we have to think more about this per Sam's question, although I'm pretty sure I'm correct, but I'll mull this over. But our conclusion was that the typical genetic nurture estimates we see in the literature could also be due to stratification associative mating. So I think the fact that they could is well understood, but the fact that they probably are is what these results suggest when we contrast the same estimates in the Korean adoptees. Of course, that doesn't say there's no genetic nurture whatsoever. It just means that the genetic nurture effect are probably quite smaller than what's typically found in the literature if we take these results at face value. And of course, I do expect there's genetic nurture effects because otherwise it'd be surprising that you have such large C-squares for many social scientific traits if there were no genetic nurture. But in any case. Okay, next section. This is going to be the G by E section, the interactions between nurture and nurture. I'll go quickly over this slide because I think most of you here know about G by E and why it's important, but still, understanding how genes interact with incentives in the economic, social, and family environment may help us understand the human capital production function, intergenerational mobility, etc. And it may also yield knowledge of the differential treatment effects of policies as a function of the individual genotypes, which may in turn lead to personalized, targeted policy intervention that provide the right environment to help individuals reach their full potentials. Of course, this raises lots of ethical issues, and bioethicists are, busy thinking about these. So, want to be mindful of these. But still, I think with people's informed consent and proper thinking about these issues, I think this could be done later. There's, for instance, there's this child's orchid theory, the orchid child theory, which if you believe it, would say that some children have genetic disposition which makes them a bit like orchids. They can bloom and be very beautiful with the right environment, but they're more fragile. Whereas you have other children who are more robust, but they don't become as beautiful as an orchid, or something like that. Now, I don't know if I believe that, but the point is, it's an empirical question, and G by E studies can help us look more into this. However, the existing empirical literature on gene environment interaction has severe limitations. Well, first, as you probably all know, until recently, genetic variants analyzed in this literature were very small. Instead of scores, they used like false positive variants typically, so that was not good. But something that's less commonly understood is that environmental variation is not exogenous, meaning they do a G by E variable, but the E variable is correlated with the genes of the individual. So for instance, if you look at the famous papers by Avshalom Caspi in Science 2002, 2003, yes, two basically, the 2003 one looks at as nearly 10,000 sites on Google Scholar, so hugely influential, but he's looking at here the G variable is the 5-HTT gene, okay, maybe a false positive, but suppose it's actually a true positive. It's still the case that the environment he's looking at, the E variable, is life stress and depression, right? And he's looking at the interaction between the 5-HTT gene and life stress and depression on, I'm sorry, looking at an interaction between the 5-HTT gene and life stress on depression. Well, the problem here is that the E variable, being life stress, is not an exogenous variable. It could be, it's not as though life stress is randomly allocated to people. So life stress is probably quite genetic as a variable. And so it's not clear that the interaction they find is a G by G or a G by E, right? So that's always an issue. Also, it's often the case that people like to do G by environment, looking at parental environment and children gene. But again, the parental environment is correlated with parental gene, which is in turn correlated with the children gene. So it's hard to interpret what's going on as a G by E rather than a G by G. Now, other limitations of G by E studies include the way the control variables were used. So there's a small paper by Keller that I'll talk about next slide about this. The variables don't have natural scales. I'll talk about this a bit later also, and then statistical power. So we addressed here all four of the first of the 5 limitations. The only thing we can't really address is statistical power because we have a small sample size. That remains a limitation of the study. All right, so in terms of estimation, what we do here is we regress various adoptee outcomes on family SES and an outcome-relevant PGI. So basically, if the outcome is cognitive performance, we can use the score of cognitive performance. If the outcome is height, we use the score of height, and so on. And their interactions and controls. So basically, regress Y on SES, PGI, and the interaction of PGI versus by PGS. And then this is the thing I was saying about controls. We include the controls as well as the control interacted with SES and the control interacted by PGI. Probably many of you have seen this already. If you're curious about why we do that, you can check that small paper by Matthew Keller. I think it's 2000, what did I think it's 2014. Anyways, Matthew Keller has a small paper saying why we have to do this when we look for interactions. Okay, so there's a gene-environment interaction when beta-3 is not equal to 0. So in economic jargon, if beta-3 is greater than 0, then the PGI SES are technical complements in the human capital production function, meaning the marginal rate of return of the PGI is increasing as the SES increases, and vice versa. And if it's beta-3 is less than 0, then they're called technical substitutes in economic jargon. And in terms of interpretation, the coefficient of PGI depends on the SES. So I mean, if beta-3 is not equal to 0, the interpretation of the specification can be twofold. You can rewrite the top part of that equation as y is equal beta naught plus beta-1 SES, and then you group the terms that are multiplied by PGI, and you get that beta-2 plus beta-3 SES becomes the coefficient on PGI. So in this case, the coefficient on the PGI depends on the SES, and in interpretation 2, the coefficient on SES depends on PGI. So here we just group the terms this way, and instead we see that SES depends on PGI. Now these two interpretations are not statistically distinguishable, so that's why we talk about G by E interactions rather than E SES modifying the PGI or vice versa. Okay, here are the results in the sample of Korean adoptees. So we have 7 different models here, 7 different outcomes, with each outcome having the most relevant score, except that for cognitive performance, we did this with both the score of cognitive performance and the score of EA. The reason is that EA is highly correlated genetically to cognitive performance, and because the score of EA explains more. It's based on GWAS of 1.2 million people versus cognitive performance. So, okay, so the bottom line here is that we let's focus on on cognitive performance as an outcome, because that's where the most interesting results are. So first, we see an effect of PGI on cognitive performance, whether we use the PGI of EA or the PGI of cognitive performance. Second, we also see an effect of family SES on cognitive performance. And third, and interestingly, we find, so this is not surprising, that's as you'd expect, but interestingly, we also find that the PGI negatively interacts with family SES. So this is only marginally significant when we use the PGI of cognitive performance, but it's very significant. The Z-stat here is greater than 3, I think, when we use the PGI of EA. Now, this is model 1 without the interacted control. So basically, going back to this, it's basically this model here, up to B4. We don't include B5 and B6 in model 1. The reason we do this is because this allows us to report the coefficient on the PGI and family SES. Once you include the interacted controls, a la Keller 2013, then you can't really interpret the coefficients on PGI and family SES without taking the average value of all, taking into account all the interacted controls. So here for model 2, we just report the interaction estimate. As you can see, it's still significant in both cases, although only marginally so when we use the PGI cognitive performance. I should mention this. It's kind of annoying. There's only 1 star here, although you have to be careful with those type of focusing too much on this level of this type of inference. But with another score of cognitive performance that's almost as large a sample size, it becomes significant at the 5% level. But here we're not p-hacking, so I'm reporting the one with the score that has slightly larger sample in the GWAS. But anyways, okay, so we have suggestive evidence here of a G/E interaction between the score of EA, or the score of cognitive performance, and cognitive performance. I'm sorry, NSES on cognitive performance. Okay, okay, I already talked about this. To visualize these results, here's, if we look at the raw data minus the controls, what we did here is we split the sample into 4 quadrants. So these are for the Korean adoptees. We split it based on the EA PGI, whether they're above or below the median, and family SES, whether they're below or above the median. And then we plot, we remove the control variables, we partialed out the control variables, and we remeaned at the mean with controls for everyone, and then we took the mean for each quadrant of cognitive performance. And so with this negative interaction, we're estimating, another way to visualize the negative interaction we're estimating is this graph here. So it appears that if you have a PGI that's less than the median, then family SES matters. Basically, moving you to the higher SES does have an effect on your cognitive performance. And if you have a PGI, and likewise, if you have SES that's less than the median, then having a higher PGI matters. However, once you have either a high PGI or high family SES, the effect of the other variable is much smaller. So that's basically kind of a saturating effect. As long as you have either a high PGI or high family SES, then you're fine, so to speak, in terms of having a high cognitive performance. Now, at least if you take the data, the results at face value, that's what this suggests, okay? What's surprising about this is that it's surprisingly robust. I was a bit skeptical of these results, I still am actually, because they need to be replicated. I'll talk more about this in a second, but these results are surprisingly robust. So here's a bunch of robustness checks, and here there's 2 panels. Panel 1 is model 2. So the model 2 is the model with the interacted controls. So that's the kind of tougher model where you interact all the controls with SES and the PGI. So only in this table, we only did this tougher model with the interacted controls following Keller. And here we have 10 different regressions times 2, because we do this once with the PGI of cognitive performance and one with the PGI of EA. And let's focus on the nicest results here with PGI of EA, although the results are similar here with cognitive performance. So with the PGI of EA, we have 10 different regressions. First, here's the baseline. So that's the same coefficient as was in the previous slide that I showed you. Now we look in males only. It's very significantly negative. In female only, however, it's only marginally it's not significant, but it's still negative. But that's the only one of all the robustness checks that's not significant. If you split the sample by age, it's significant in both cases. Then I was talking about one of the limitations in G/E study is the issue of scales. So for variables like cognitive performance that don't have a natural scale, You could induce a false G by E by simply taking the square of the variable, right? If you have a simple model where it's cognitive performance equal to gene plus environment and no interaction, and you take the square of that, because you want to do it to scale transformation, then you'll end up with a square term, an interaction term between genes and environment on the right-hand side, right? So to check whether the model is, the results are robust to scale transformation, here we took the square of cognitive performance instead of just cognitive performance. And here we took the square root of cognitive performance. And again, things are still significant. Of course, the coefficients are very different in scale because, here it's cognitive performance squared versus square root. We did the same thing for SES. We changed the scale to see if this is robust, and results are robust to change of scales in socioeconomic status. And here in column 10, we use an extensive set of controls that include the baseline set of controls that are age, sex, placement age, and PCs, and what else? There's something else I'm forgetting, but anyways. As well as, and then we added to these the mother and the father's birth years, the number of adoptive and biological sibling the adoptee has, and dummies indicating whether the adoptive parents reside in this city or suburb, and whether they are still married at intake. So we just use a bunch of additional controls, and it's still robust to that. Okay, now this seems quite robust, but I'm skeptical of this result still because we only have 361 genotyped adoptees. So, and I've been critical of false positives. So try to do the responsible thing and think, what's the power we have, the statistical power? So we derived an analytical expression to calculate power under simple assumptions. And the adoptees, G and E, are uncorrelated. So it turns out basically power is mainly a function of that's what I wrote here. So the adoptees, G by E, are uncorrelated. So power is very easy to calculate. It's basically a function of the unknown R squared of the G by E interaction and the number of degrees of freedom. Right, so there's some other parameters in it, but really what drives this power thing is the assumed R square of this interaction, as well as number of degrees of freedom of regression. So we know everything. The only thing we don't know is this here. So we need to assume various values and see what we get in terms of power. We also verified these power calculations through simulation, including simulations that model the family structure of the data. The conclusion is the statistical power could be limited to estimate the significant G by E effect in our sample of Korean adoptees. However, this depends really on what you assume is the true R squared. So if you assume that the R squared of the G by E correlation is 1%, then power is 43%. And we're kind of in trouble. 43% is not very good power. If we assume that it's 0.5%, then power is 24%. We're even more in trouble. However, if you assume a G by interaction that's 2%, then power is 81%. So I think actually this should be, I think that's a typo. I think it's supposed to be this, but anyways, I should double check. So what's the right R squared to assume? Well, who knows? But by comparison, the R squared of the PGS of EA, just linearly without interaction, is 5%, and the R squared of the SES variable is 2%. So, what's a reasonable assumption for the R squared of the interaction of these two variables? I don't know. It's probably, my sense, it's probably more around the lower end. So I do think we're underpowered, and I do think this may be a false positive, but, I do think this will have to be replicated in an independent data set, which we don't have. But I think it's still nice to report the results with the data, even though we think we're underpowered, but with proper caveats. As an illustration of what I think is, would be the ideal setting where you have quasi-random or exogenous environment relative to the genes, for G by E study. Okay, any questions on that before I conclude? Okay, so then to conclude, I should mention also, maybe I have a few minutes, I'll go here. We have this slide here where we did the same G by E specification, the sample of white non-adoptees, and we don't find anything significant in terms of the G by E interactions here. So, this could be, there's two possible reasons for this. One reason is that the results in the Korean adoptees were false positives and they don't replicate in the white non-adoptees. But the other possible reason is that because in the white adoptees, family SES is not exogenous to the score of PGI, that this we have some, we explained the math in the paper, but basically that these effects cancel out, that there's a true, it's possible as a true negative G by E interaction, but there's also a positive G by G interaction, and that the two cancel out when we use non-adoptees, basically. Okay. Okay, concluding remarks. So we analyze a unique data set of genotype adoptees who are quasi-randomly matched to adoptive families. Analysis with the pedigree data, meaning the ACE model, and molecular genetic data both indicate a role for genetic and environmental factors, though family variables tend to be less significant. So there's evidence of that a large part of the typically estimated indirect effects of genetic nurture is driven by stratification or confounding. So that's our best interpretation of our results that we presented about genetic nurture. And then interaction analysis suggests that genes for EA, quote unquote, meaning a polygenic score for EA, and family SES are substitutes in the production function of cognitive skills, but replication of this result is key. And then my usual concluding pitch, although I think most people here are already convinced, it's likely we're on the cusp. I think we're in the middle of an explosion of discoveries in geneconomics, or social science genetics, I should say. As available data sets are multiplying, sample sizes are rapidly increasing, and the predictive power of polygenic indices for economic preferences and outcome and social outcomes and so on will rise substantially with many applications. And I should add another point now is there'll be more direct effects estimates, which will allow to create scores with direct effects only. So I'm very excited about all this. Okay, thank you. Let me know if you have any questions.
Thank you, Jonathan. Let's go to questions for the remaining few minutes. Maybe while people are thinking, I had one curious result that you had, Jonathan and Lauren, was that for the adoptees, that mom's EA, you could either call it 0 or negative on adopted child's cognitive performance, if I remember correctly. That mom's EA-PGS, that seems like a strange finding. I'm wondering if you have any thoughts on the meaning of that finding.
Are you referring to, just to make sure I understand, are you talking about this finding here?
It's that one plus the next one when you take out dad.
Yes,
the next,
yes.
yeah, that's right.
Yeah. Well, so my interpretation of that is that, so first we do find, to use this, the first thing I'll say is, like, let's focus, for instance, on this effect of going to college as the outcome, because I think I can make a good parallel with earlier results. So regressing going to college on the mom's EA-PGS is just not significant here, even though there's 301 individuals. So, however, earlier, we saw that going to college here is positively correlated with family SES. And the reason we didn't include mom's EA in this regression is because EA is a component of SES and become correlated. But if you include EA instead of family SES, you'll have a positive sig coefficient here, okay? So we do find a treatment effect of mom's EA and whatever is correlated with it here. We have to be careful with the interpretation. On going to college. So the fact that we don't find this here is quite surprising when we use the score of EA instead of the mom's actual EA. And my best interpretation, my best guess of what's going on here is just low power. So I do think there's, my best guess is that if we had a larger sample size, we had more precise estimates, we'd find genetic nurture here. It's just they would be smaller than the genetic nurture we find when we have non-adoptees. So that would be my best, but who knows? It could also be that there's really no effect of the PGS of E on going to college, even a large sample with precise estimates, but I'd be very surprised if that were the case.
Yeah, I have a quick follow-up on this point. You phrased some of this as a stratification, which sounds like a bit of a technical artifact, but I wonder if it's also possibly something more mechanistic. For example, is it possible that genetic nurture need to happen at a much younger age, even before these kids are adopted? So I wonder if you have, for example, age at adoption data in this, and for example, stratify kids into different age groups to see if this is a consistent pattern?
Yeah, that's a very good question. I think that's the takeaway from this presentation, Lauren, is we really need to clarify the framework to our terminology to talk about pre versus post-adoption timeline. So yes, you're absolutely correct that a real test of genetic nurture here would be to have to quasi-randomly place the Korean adoptees at conception. So I guess with in vitro fertilization, well, they're not quasi-randomly placed, but like, you know, if you had, think of an experiment that I'm not saying would be ethical or anything, but like a theoretical experiment, you would want to quasi-randomly place the embryos in various mothers, I guess, at conception, right? And then you could really see here, you could really have this genetic nurture estimate that's clean of the stratification. So yeah, you're fully right that this could be that there's genetic nurture happening pre-adoption. Yeah, and you're right about it's a good idea to check, to split. We have placement age, so we could check whether we see any difference between those that are adopted, let's say before 6 months or before a year versus after. Everyone's adopted between 0 and 2 years. But the main issue here is we may have too little data to find anything precise. Yeah, that's a good point.
I think the other issue too, and speaking to it's what you mentioned earlier, Jonathan, and it might also speak to some of these lack of correlations in the adoptees, is the range restriction in the parents' polygenic score. So it could be that we have a much, nicer distribution in the non-adoptees, and that's why we're able to see, the associations more. And then when we're looking in the adoptees, we have more range restriction in the polygenic scores. So that could be another, and yeah, we should look at that more as well too.
Yeah, absolutely.
Jonathan, so I'm just thinking out loud. So I'm not really sure if this makes a lot of sense, but I'm just wondering if this negative interaction effect that you're finding may be just a statistical artifact of measurement error in the two variables that you have. So suppose that you have, so you have a measure of parental SES and you have a measure of child's genotype.
Yep.
And both of them, they may be a combination of true signal plus some random noise. Mm-hmm. And if you combine the two, so if you interact the two and you have a pair where the two variables were both just influenced by a random positive noise shock, wouldn't you expect that this sort of leads to some sort of regression to the mean, so back to the actual signal?
So you mean for what coefficient in particular? You mean, for instance, this one here? You're saying
the interaction effect that you had earlier.
Interaction effect. Yes.
Yeah.
I was still in the wrong mindset when listening. Yes, certainly. So if you have the PGI with noise, which obviously PGI have noise because by definition they don't explain, they're not the perfect additive snip factor. And family SES is probably noisy too, almost. I mean, like certainly. You're fully right. So there's an attenuation bias here going on for sure, and that will, all these non-significant results here could be due to that plus the small sample, right? Yeah, it's a fair point. And it's kind of made worse when you multiply two variables, right? Because you have attenuation bias of both. Yeah, that's a fair point.
Well, we're right at time, so I really appreciate Jonathan coming to discuss this great paper of his and Lauren's and co-authors with us today. And he has more meetings today with us, and we're really pleased that he'd share part of his afternoon to further discuss these. So thanks very much for coming, Jonathan.
Well, it's my great pleasure. Thanks very much for having me. And actually, you made some very good comments, so that's good. Okay, thank you, everyone. Bye.